We are right at noon, so let's go ahead and get started. Hi, I'm Dr. Stacy Wall. I'm the liaison to the Basic Sciences and the Health Sciences Library here at VCU, and this is the How To Talks by Postdocs, a series where we have postdocs come and teach us about something related to their research or their career, and they teach us how to do it. Okay, today we have Dr. Apoorva Marie, and she is a postdoc in Dr. Ian Morgan's lab, and she's going to start her talk with an introduction about her background and how she really got to where she is, important to her how-to topic. So I'm going to let her do that for us, okay?
Thank you, Dr. Wall. I want to start off by thanking you all for taking your time to attend today. In today's talk, I will cover about how to make an in vitro model more applicable to the clinic, or in other words, say how one could warrant the translatability of your own project to the clinic with having a right model system. So a little about me. My name is Apoorva Marie Prabhakar. Hi again. I got my dental degree from India. Then I moved to Louisville to pursue my master's in oral biology. My thesis there focused on studying about the oral periodontal pathogen called the Porphyromonas gingivalis. Then I recently graduated in May from VCU with a PhD in oral health research from the dental school. My dissertation during my PhD focused on studying the human papillomaviruses and deciphering the molecular mechanisms of HPV-induced carcinogenesis. Currently, I am continuing as a postdoctoral fellow in Dr. Ian Morgan lab, studying HPV as well. So what do I bring to this virtual table with regards to the in vitro cell culture model? I have been working with the in vitro cell culture models to dissect the human papillomavirus life cycle for about 4 years. That's during my PhD and continuing during my postdoc now. In our lab, we have been able to adapt and make use of these models to study HPV and cancers. In today's talk, I will discuss how to adapt and apply the in vitro cell culture models that better mimics disease processes and make it more applicable to further clinical studies. So an outline of today's talk, I will start off with a background, giving an overview of different model systems there is out there. Next, I will talk about why selecting the correct in vitro model system is so crucial. And I will also talk about how to make an in vitro model system into better mimicking the disease process that is about how to make it into a disease model, and what factors goes into making these models, and what factors should a researcher consider when selecting a model system to study a disease process, and why is it important to take an integrated approach, how to best go about building the right biological model system. We'll cover that as well. And we will finish off by talking about different shifting of the trends and the perspectives involved in these in vitro models, and talk about the customization, how you can make the in vitro model your own, and what can researchers do to warrant the translatability of the system. And we'll finish off with some thoughts. Since we have a diverse audience in our talk today, I will go through a brief background that covers relevant aspects that set stone for today's presentation. So simply put, a biological model is a surrogate for a human being or a human biological system. And these are invaluable to provide new insight into understanding of the normal and as well as the abnormal functions from basic gene level to the phenotype level, and also to provide a basis for preventive and as well as the therapeutic intervention in human diseases. As you can see here, there are so many different models which are used for this purpose. With so many model systems to choose from, deciding which will work best for your research can be daunting. At the present time, animal models of human diseases to in vitro model system are significant tools widely used by the biomedical research community. Today, we are just focusing on the most widely used models, that's the in vivo and the in vitro model. firstly, let's identify the difference between the in vivo versus the in vitro system. In vivo is Latin for within the living. So it refers to the tests, experiments, and the procedures that researchers perform in or an all living organism, such as a person, laboratory animal, or even a plant. Whereas in vitro in Latin for is in glass. In simpler terms, it describes the experimenters as a researcher perform outside of a living organism. An in vitro study occurs in control environments such as a test tube or a petri dish. So a right in vitro model can be a robust alternative to using animals. So adapting and applying a right in vitro model to your experiment is very crucial for this purpose. So we'll start off with the cell culture, which is a type of in vitro model. So what is cell culture? Cell culture is the process by which cells are grown in a controlled environment or a condition outside of its native environment. So cell culture provides an invaluable complement to in vivo experiments. So why do we use cell culture? As I mentioned, cell culture is one of the major tools used in cellular and molecular biology. It provides an excellent model system for studying the normal physiology and the biochemistry of cells, the effect of different drugs and the toxic components on these cells. And also we can study about the carcinogenesis process using the cell culture model. This model is also used in drug screening development and also for screening of large-scale manufacturing of biological components like vaccines and different therapeutic programs. So cells, they are the fundamental unit of life. And as I mentioned, they're an incredibly useful tool in every day in the biological research process. So this is something we use every day in our lab as well. So to briefly describe how we go about cell culturing, so the cells used in this model may be removed from the tissue directly, and they can be disaggregated by enzymatic or the mechanical means before cultivation, or they can be derived from a cell line or a cell strain, which can be commercially acquired, and later on you can establish these cell lines. So firstly, there is the primary culture, which refers to the stage of the cell culture after the cells are isolated from the tissue and allowed to proliferate under the appropriate condition until they occupy all the available substrate, that is, they reach a confluency, which is required for your study. At this stage, the cells have to be subcultured or passaged by transferring them onto a new petri dish or a new vessel with a fresh growth medium to provide more room for the cells to continue to grow. As I mentioned, you can acquire these cell lines as well. That is, after the first subculture, the primary culture becomes known as the cell line or subclone. So the cell lines derived from the primary culture have a limited life, that is, they are finite as they are passage. Cells with the highest growth capacity predominate and resulting in a degree of genotypic or the phenotypic uniformity in these populations. So this is a very basic overview of what cell culture technique is, how we go about doing the process. So I'm not going to talk about the process or how we do the basic cell culture. Today, we are going to talk about why selecting the correct in vitro model system is so crucial for everyday research. So in vitro research has many advantages over in vivo research, such as you can have a tight control of the chemical and the physical environment. It's reduced cost when compared to housing animals and doing your in vivo studies. And also you can have high throughput using this in the in vitro system. And also, as I mentioned, you can have reduced animal use. However, one of the substantial weaknesses of in vitro experiments is that they fail to replicate the conditions of cells in an organism. Example, as I mentioned, the isolated and the cultivated primary cells usually differ strongly from the corresponding cell type in an organism, limiting the value of in vitro data to predict further the in vivo behavior. To get better out of the system, one has to bridge the gap between these both in vitro and the in vivo system for better translation of research. And it is thus essential to adapt to a better model system, especially the better in vitro system, which can account for this translation of your research. So how can we overcome these shortcomings and how we can make an in vitro system into a disease model? We can use we can do this by use of the 3D cell culture system, which can literally add a new dimension to this 2D in vitro cell culture system. So in 2D cell culture system, cells are grown on a flat dishes with a coated surface to help them adhere and proliferate. These models, as I mentioned, are not representative of the in vivo cell environment, as they do not mimic the microenvironment found in the body tissues. The use of 3D cell culture refers to the process of growing cells in their native context, that is the in vivo environment. This is done by adding a third dimension, which can mimic the complex interactions between the cells, as well as between the cells and as well as the cells and the extracellular matrix, or the ECM. In contrast to the monolayer or the 2D cell culture, where cells are mostly exposed to the media and the plastic in the cell culture dish, in 3D cell cultures, all the cells in the culture are exposed to either to other cells or, as I mentioned, the extracellular matrix. Cell cultured in the simplistic layers lose their original phenotype and functional characteristics, often becoming de-differentiated and are very distinct from the tissues that they are originally derived from. Hence, the biological response to these cells may not be physiologically relevant, which can be a major factor hampering the predictability and thus further in the utility in, say, drug discovery. Due to their architectural similarity to in vivo tissues, 3D cell cultures retain the phenotype and functional characteristics of their in vivo counterparts, thereby providing a more physiological relevant in vitro system to evaluate different biological processes. So even the 3D cell cultures are very fastly evolving and are fastly replacing the 2D cell cultures due to their reliability as an in vitro approach to predict the in vivo responses. So today we'll focus on how we can adapt this 3D cell culture in your experiment. So next, I draw on my experience to highlight the importance of making use of the right in vitro model that can potentially provide successful translation of the basic findings to the clinic. As mentioned, in Morgan Lab, we study the human papillomaviruses, and we dig deep into exploring how they cause cancer. So briefly describing, broadly, the viral infections are responsible for causing 15% of all human cancers, and HPV accounts for more than 600,000 new cases of cancer worldwide annually, and the number is just increasing. HPV can infect both cutaneous and the mucosal epithelium. As you can see from the graph here, HPV causes a wide variety of cancers, both in men and women. And HPV is mainly responsible for causing cervical cancers in women, and in men, HPV infection is predominantly the causing organism for the oropharyngeal cancers. So HPV, the human papillomaviruses, are small, circular, non-enveloped, double-stranded DNA viruses, which has a small genome of about 8 kilobases in length. The high-risk HPVs, the HPV 16, 18, and 31, are responsible for causing cancers, whereas the low-risk HPVs, such as the HPV 6 and 11, can cause genital warts. Research has shown a strong correlation between the oropharyngeal cancers and HPV, with 90% of HPV positive cases primarily caused by HPV 16, that's the high-risk HPV. Therefore, it is essential to understand the HPV life cycle and the interactions between HPV and the host, which can provide insights into new targets for infection control and also allow us to formulate novel targeted therapeutic protocols. So HPV's life cycle depends on the host differentiation process. It occurs in 4 different phases. The first phase is called the establishment phase. In this phase, the virus gain entry through the microabrasion in the epithelium to infect the basal layer cells. After the successful establishment in the basal cells, the HPV is maintained in an episomal state, in other words, as a circle at a copy number of about 50 to 100 copies per undifferentiated cells. Low-level expression of the viral early proteins, such as a viral protein called E1, E2, E6, and E7, occurs in this phase, which helps in the evasion of the human host immune response to the infection. Following this, during the maintenance phase of the viral life cycle, HPV-infected basal cells undergo differentiation. And the HPV genomes, as I mentioned, are small. They do not encode for polymerases or other enzymes which are necessary for the viral replication. Thus, HPV relies on the host cell replication proteins to facilitate their own viral DNA synthesis. As the infected host cells migrate towards the upper layers, different viral proteins are expressed at higher levels and viral DNA is amplified. This is the amplification stage of the viral life cycle. In this amplification phase, the number of copies can range of the viral copies can range from up to thousands per cell. Subsequently, in the final stage, the viral genome gets packed into a capsule to form a progeny virions. This shed viruses can then initiate a new infection. Thus, as you can see, there's an intimate relationship between the HPV and the epithelial differentiation, which is brought about by expression of various viral proteins by this crosstalk between the virus and the host. So this just establishes the importance of studying the virus and the host as a component and not just studying the virus on its own. So this is briefly about the virus life cycle. So next, let us explore how using the right model system be helpful, especially while studying this myriad of host pathogen interactions associated with disease like HPV infections. So although the conventional monolayer cell culture system can be employed to investigate the functions of the individual viral proteins in different transient functional assays like the transcription or the replication assays, which we use in our lab, or also in investigating the immortalization functions of or immortalization status of these HPVs by using a Sudden blot. The attempts to produce human papillomaviruses using this conventional monolayer cell culture has been unsuccessful. For many years, the field of HPV research was hampered by the lack of tissue culture system that allowed the full HPV life cycle. Advancements are actively made in the field of tissue engineering regarding the development of the system called a 3D organotypic model of associated HPV diseases, wherein we are developing different models where we can study the whole HPV life cycle, focusing on the carcinogenesis process rather than just growing them in the 2D cell culture. One such advancement, as I mentioned, is the development of organotypic rat culture. As I mentioned before, due to the correlation between HPV life cycle and the squamous epithelial differentiation, the most powerful in vitro models are represented by these 3D organotypic epithelial tissues or the rat cultures. So we can basically recapitulate the viral life cycle by growing the cells in these rat culture. So this is done by seeding the cells onto a collagen plug and which contain the fibroblasts and allowing a monolayer of keratinocytes to grow, and then lifting this plug to grow at the air liquid interface on a metal grid as a support. And at 13 days, these cells are fixed and sectioned, kind of like a biopsy. And this we can subject to H&E staining and check for different tissue architecture. As you can see here, this is the normal keratinocytes, which closely mimics that of your own epithelial tissue on your skin or in your oral cavity. This is epithelial layer on top of a collagen or the fibroblast layer. And then with the HPV containing cells as well, you can see, you can compare different architectural of these tissue and use it for your study. So in this way, raft culture has allowed normal keratinocytes to stratify and fully differentiate, as I mentioned, in a liquid air interface culture to produce the full thickness epithelial tissue when feeding on top of a dermal equivalent consisting of a fibroblast layer, or in this case, a fibroblast populated collagen gel. So these 3D models have provided us an environment which permits for recapitulating and modulating the infection program of cancer-associated high-risk HPVs, which, as I mentioned, cause majority of cervical as well as the oral cancers. This is also further helpful to study the crosstalk between the stroma and its microenvironment on the HPV-infected epithelium. So not only HPV, most recently, the system has been used for the study of other viruses like herpes simplex virus infection as well. So from previously published papers from a lab, such as here, we can observe that the productive viral life cycle can be recapitulated fully in these keratinocytes isolated from, say, anogenital or the oropharyngeal tissues that can harbor the circular HPV genome when grown in these raft cultures. These raft culture provides us with significant insights into the molecular biology of the HPV life cycle, and as well as the pathogenesis of, we can dissect the pathogenesis of how these HPVs cause cancer. So these 3D epithelial models also can further provide us about different vital platforms for screening of different antivirals, as well as testing novel therapeutic compounds as well. One of the many advantages of using raft culture, especially in our study, is the ability to interrogate the virus-host interaction in these stratified epithelium in these individual cells by using in situ assays of thin section of formalin-fixed paraffin-embedded epithelial tissues. Conventional biochemical assays can also be conducted on the epithelial portion of the raft cultures, including the viral isolation and titering by, say, qPCR, the quantitative PCR, and also we can isolate the protein for immunoprecipitation or co-immunoprecipitation or Western blots using these raft part or the epithelial part from these raft cultures, and also nucleic acid extraction for Southern blots or many other functional assays as well. So in this way, these 3D epithelial models is very useful in our research. So this is just one example of how one can adapt and apply the in vitro system to get the most of it to study a disease process. So there's an increased acceptance of the in vitro, which can rely on the robustness of its robustness, and we need to improve on what we have and adapt, as I mentioned, which can help us further to reduce the uncertainties of using this in vitro-based predictions. So it is crucial to identify various factors that needs to be considered while we choose a model system. So to develop the right model system for research, one needs to crucially consider what is the purpose, what's your research question. So first we need to identify the purpose of your experiment. Currently, there is no single model which can capture all of human biology. So the first step is to know what physiological process are most important to have in a model in order to establish, say, whether a drug is effective and safe in your drug screening if you're in cancer research. So that's your first important step. So the next step is to determine how to capture those processes in this in vitro model system. So there should be a confidence in the correlation of the results from your in vitro model system to that of the human disease, and that can be achieved by having a good in vitro model system. And also we need to determine the extent of the factors like the intrinsic and the other extrinsic factors which can have a potential effect on your model system. So considering all those factors is also very important while you're developing a model system. And also you need to identify not just the pros, but also what are the limitations of using a different model systems before you use it for your study. So having checking all these off while developing a model system is very important when you develop any model system for your study. So it is very vital to take an integrated approach and to identify how to best go about building the right biological model system. So taking an integrated approach is key in this process. So we start off by, as I mentioned, identification of the question. A good model design defines the question to be answered by the model, determining the desired resolution from microscopic to macroscopic level, given the complexity of the biological system, say from the molecular level, even to the population level of a study, will aid in outlining the scope of the model system you're using. And also it will have an effect on the expansion of your study, which will determine different outcomes from your study. So that's your first step, identification of the question. Next is model building. To build a model, a physiologically and a pathologically relevant system that should be tailored to make whatever you observe to expand it to the wide scope of your study. So the driving factors of the system, which is consistent with the parameters of the questions you identified in your first step, is very important. And you should be able to manipulate these factors while utilizing it in your system under experimentation. And also your system should produce a relevant as well as a useful readout that properly addresses the endpoints of your study. So that is very crucial when you are building a model. Next up comes the model testing. So testing of the model involves adjusting the parameters of the driving factors as input or generating the outputs that allow the evaluation for comparison for the real system. So that is your disease model. So this is again very crucial while you're actually building your model, and you need to identify all the different factors and engineer have good controls and have good statistical power to your study so that we have better outcome from your experiments, which can have an effect later on when you apply your project to a clinical level. Next comes the outcome evaluation. So the endpoint of the study should be compatible with that of the real system, allowing the comparison between two systems at a translatable base. So whatever you do in your lab, you should always have an eye out of what's the translation of your research. So that's gonna help in developing better model systems. So this will help in better clinical outcomes, and this will also help in resolution of a better model from your research. Next comes the model improvement. So you need to be continually assessing your model system and get more information by keeping an eye out on what's happening in different clinical trials from the basic research and trying to fill the gap, what is there between your basic research to the translatability of into the clinical research. So this is a continuous cycle when developing a model system. So while I mentioned the primary benefit of 3D cell culture gives us a greater resemblance to the in vivo conditions than its 2D counterparts, it is undoubtedly valuable in physiological research, but 3D culture are still more difficult to use compared to the 2D cultures. But there are increasingly more innovations made towards improving these 3D cultures to be used in a very large scale. Today, a variety of 3D culture models ranging from use of the scaffolds to use of the hanging drops and spheroids makes it easier to choose a different type of in vitro 3D model for using it based on your research goals and experiments. So choosing the right in vitro biological models lays a strong foundation for the entire research project. So it is important to customize your research model in order to give a better clinical translation of your project. Is very key when you're developing a research in vitro system. So instead of the conventional notion of bench to bedside, start from the bedside. So think of what required at the clinical level, and you can use that while developing your research and addressing these problems. So think about it as a bedside to bench approach. So it goes hand in hand, and having this integrated approach while building your in vitro biological model system, as a researcher, one can achieve more reliable results and also will help in the long term, saving time and also lowering costs. So thank you for your time. I'll take any questions you guys have and go from there.
So I have a question to get us kicked off. So when you're thinking about staying connected to what's happening in the clinic and how that affects your design of research models, how do you keep track of how do you find out what's happening in the clinic?
So usually as basic researchers, we are not kept aware of ourselves of what's the clinical translation. So there are so many clinical reports which are published. So not just the basic research papers, we need to keep an eye out on different clinical reports or clinical trials which are happening out there, which basically starts from the bench aspect of it. So keeping yourself aware of those as well is going to help with that bridging that gap between your bench to the bedside aspect of it. Thanks for your question.
So do you have like a favorite clinical journal that you'll browse, or do you use like PubMed? How do you find stuff related to your project?
I usually use PubMed. I go through PubMed and see what's the latest research out there, and be it oral cancer research or be it the cervical cancer research. I just keep an eye out on what's happening at the clinical trial level and try to adapt and see what's lacking or what's the gap between what I am doing as a basic researcher and what's happening out there at the clinical level. Any other questions?
Do you have ideas about, hi, this is Pam. Do you have ideas about how you could see the integration between the clinical and the basic research moving forward? I know that it seems like you do a lot of work on your end. How do you see that moving forward?
I guess we still see a gap between as a basic researcher and the clinical researcher, but there's so much potential and steps we can take towards bridging the gap. So I would say it's going in a positive direction, but there's still such a gap between that needs to be filled. So hopefully it's going to improve soon.
Thank you.
Thanks for the question. Okay. Is there any other questions?
All right, everybody, before you head out, I'm going to post a link to an evaluation in the chat real quick. That's for you guys to tell us what you liked about this talk, what you like about the how-to talks, and to provide Dr. Tati Marie with any feedback on this topic or anything else that you'd like to hear about. So thank you all so much for coming. We have one more talk in our How-To Talks by Postdoc series next Monday. That'll be Bayesian statistics with VCU Science Communication Award winner, Dr. Sean Cobley. Okay, so we look forward to seeing you then. Thank you again so much for this excellent how-to talk, and enjoy the rest of your day, everyone.
Thank you.
Thank you.